T and L biotechnology, cord blood NK cell culture workflow. Coatings of culture flask. Pipette 9 ml of DPBS into a 50 ml centrifuge tube. Add NK reagent to the tube and resuspend using a pipette to mix thoroughly. Slowly add the DPBS containing reagent to a 75 cm squared culture flask. Secure the cap and shake the flask gently on a flat surface for 30 seconds to 1 minute to evenly distribute the coating solution across the bottom of the flask. Further incubation conditions: coat at 4°C for at least 18 hours or at 37°C for 2 hours. Transfer the cord blood from the collection bag to a 50 ml centrifuge tube and centrifuge the whole blood. After centrifugation, use a pipette to collect the supernatant as plasma. CBMC isolation process. Resuspend the pelleted blood cells in an equal volume of saline and mix well. Slowly layer the resuspended cells onto the fickle layer, ensuring clear separation. Centrifuge with a fast acceleration and slow deceleration. After centrifugation, collect the buffy coating. Use a pipette to discard the supernatant and carefully aspirate the CBMC layer from the interface to maximize cell recovery. Transfer the buffy coating into a new centrifuge tube and resuspend it in saline before centrifuging again. Discard the supernatant after centrifugation. Wash the cells twice with saline and resuspend them in saline or serum-free medium. Take a small aliquot for cell counting. Centrifuge to 0.5 × 10 to the 7 cells for seeding. Excess CBMC can be cryopreserved and used later for cord blood NK cell culture. One cord blood sample can be used to culture multiple NK cell batches. Seeding process. Preparation of complete activation media. For each bottle of NK activation media, add 1 vial of NK activation additive 1 and 1 vial of NK activation additive 2. Add 23.75 milliliters of NK complete activation media to the cell. Pellet after centrifugation. Then add 1.25 milliliters of HPL. Mix the cell suspension thoroughly using a pipette and add NK reagent B. Take the coated flask, allow it to return to room temperature, and use a pipette to remove and discard the coating solution. Add the cell suspension to the coated flask, gently mix, and place it in the incubator at 37°C with 5% CO2 for culture. First feeding, day 3. Gently remove the cell culture flask from the incubator, ensuring minimal movement to avoid disturbing the liquid and disrupting the activated cells. Clusters at the bottom of the flask. Observe the cells under a microscope. Proceed with feeding when the cell clusters cover more than 20% of the flask bottom. Add 23.75 milliliters of pre-warmed NK complete activation media and 1.25 milliliters of HPL, bringing the total culture volume to 50 milliliters. Second feeding, day 6. Gently remove the cell culture flask from the incubator under the microscope. Observe significant cell proliferation with an increase in medium to large cell clusters. Add 23.75 milliliters of NK complete activation media and 1.25 milliliters of HPL 5% autologous plasma, bringing the total culture volume to 75 milliliters. Subculture and third feeding, day 7. Preparation of complete expansion media, the NK expansion additive 2 and recombinant human IL-2 prot in using culture media. For each bottle of NK expansion medium, add 1 vial of NK expansion, additive 1, half a vial of NK expansion, additive 2, and 1 vial of recombinant human IL-2 protein. Remove the cell culture flask from the incubator, transfer the cell suspension from the T75 flask to a T175 flask. Rinse the T75 flask with 71.25 milliliters of NK complete expansion medium and transfer the rinse to the T175 flask. Add 3.75 milliliters of HPL, bringing the total culture volume to 150 milliliters. Bagging and fourth feeding. Day 9. Connect the culture bag port to a 50 milliliters syringe. Transfer the cell suspension from the flask into the culture bag via the syringe connection. Then, pour 142.5 milliliters of complete expansion medium into the flask, rinse the flask, and transfer the rinse to the culture bag. Add 7.5 milliliters of HPL, remove excess air, and manually tap the culture bag to ensure even distribution of cells. Finally, fold the culture bag to about 2/3 and place it in the incubator for further culture. Fifth feeding, day 11. Connect the culture bag port to a 50 milliliters syringe. Add 15 milliliters of HPL, followed by 285 milliliters of complete expansion media, and return the bag to the incubator for further culture. Sixth feeding, day 13. Connect the culture bag port to a 50 milliliters syringe. Add 600 milliliters of complete expansion medium, remove excess air, and place the bag back in the incubator for further culture. splitting and 7th feeding day 15. Connect a new culture bag to a syringe. Split 600 milliliters of the cell suspension from the day 13 culture bag into the new culture bag. ATD 450 milliliters of NK complete expansion media to each bag. Place both bags in the incubator for further culture until cells are harvested around day 17 or 18. NK Cell Culture Kit 3.0, cultivating cord blood NK cells is effortless. Cytokine-only culture, Xeno-Free, for 26 billion NK cells from a 2L system, greater than 90% purity. The blood sample can be from either fresh or cryopreserved CBMC. The initial cell count of 2.5 E7 is sufficient. One cord blood sample can be used to culture multiple batches of cord blood NK cells.